Hi everyone, my name is Tyler Roche. I'm a grad student in the HUD lab at Georgia Tech, and I'm working with the Center for Chemical Evolution. So I'm just going to thank you all for coming to my talk. I'll be discussing the prebiotic relevance of ketose sugars to the origin of aldose nucleosides. So the RNA world model is a popular hypothesis for the formation of the first genetic polymer, RNA, as posed by the model. At the CCE, we think that there could have been perhaps a Proto RNA or some sort of genetic polymer that preceded RNA in the chemical evolution of genetic polymers. So you can imagine that in the same way that you could have a modification from RNA to DNA by the removal of the hydroxyl group on the 2 carbon and the replacement of thymine for uracil, you could have had some other substitutions from a Proto RNA system, including substitutions of perhaps the ionized linker, which in RNA is a phosphate, the trifunctional connector, which in RNA is ribose, and the recognition unit components, which in RNA are the canonical nucleobases that we know. So with this model in mind, it would be interesting to understand how these components were formed and combined to form either the Proto RNA or the RNA itself as chemical evolution proceeded. So in terms of RNA synthesis in a prebiotic context, there are several stages that had to be undertaken for the RNA to form into its fully oligomerized state. So we can imagine that the several steps included sugar formation, nucleosidation or glycosylation, phosphorylation or oligomerization. And all of these steps pose various challenges to prebiotic chemists attempting to understand how these happened. So today I'm going to focus on sugar formation and nucleosidation as two of the steps that I'm interested in looking at and investigating. So the foremost reaction is the reaction that's most frequently invoked to explain the formation of sugars on the prebiotic Earth. And in fact, the foremost reaction can form a variety of different sugars. You can have branched sugars, unbranched sugars, ketose sugars, aldose sugars, all sorts of different sugars with this reaction. So if you're looking for ribose, which is part of RNA, it's not necessarily going to be the easiest thing to find. In fact, in this chromatogram, you can see that it's one of the many peaks present, and it's not particularly large as one of the peaks. So the question becomes, how did life select for ribose? How did it come out of this vast mix of sugars? Another question regarding the formation of RNA includes nucleosidation. So there have been experiments showing that canonical nucleobases don't readily form nucleosides when combined with RNA or combined with ribose and water. You can form some nucleosides with some of these, but it's not an easy process. It's not something that happens in very high yields. However, research in the Center for Chemical Evolution has looked at various non-canonical nucleobases that have various properties that are beneficial potentially to their prebiotic formation into these nucleosides, including their reactivity and their ability to undergo supramolecular assembly. So when you react these nucleobases with ribose, you do in fact form nucleosides that can perhaps be selected for into a Proto RNA system. So the non-canonical nucleobases react more readily to form these nucleosides. So research performed by one of my group members, David Fialho, included reactions with sugars and triaminopyrimidine to see if you could form nucleosides with all these different sugars. And the sugars of interest, obviously, for me include ribose and Ribulos. So when you react TAP with these sugars, you can form these nucleosides fairly readily. But an interesting result of his was that if you react Ribulos, which is a ketose sugar, with TAP triaminopyrimidine, you can in fact form nucleosides that are formed as if they're from the aldose of that sugar. So aldonucleosides but not ketonucleosides were detected as products. So to kind of get into that, talking about aldoses and ketoses, what do I mean exactly? So when you look at these two sugars, Ribulos and ribose, they're isomers. They're both 5-carbon sugars. Ribulos is a ketose because it has a ketone functional group, and ribose is an aldehyde or an aldose sugar because it has an aldehyde functional group. And in fact, these can interconvert. This is the mechanism by which it is thought that they're interconverting, and it involves the formation of an enol and then the migration of a carbonyl group. So you can have this interconversion between these ribose and ribulose sugars or any other aldose and ketose sugars. And in fact, you could have the same sort of thing happening with arabinose, which is just another isomer of ribose that differs based on the chirality of one of the carbons. So my hypothesis when looking at these sugars is that perhaps the nucleobases in solution could attack and trap the aldose sugars, but not the ketose sugars, in the form of nucleosides. And this is perhaps how the nucleosides were able to be formed on the early Earth. So for one of my first experiments, I wanted to see if I could look at the isomerization of Ribulos, which is a 5-carbon sugar that could isomerize into ribose and arabinose. And I basically incubated this Ribulos at 65 degrees for 3 days and looked at it by NMR. And so the first spectrum you see up here is just the time point 0, which should just be Ribulos in solution. And in fact, you do see the standard spectrum for Ribulos. And when you allow that Ribulos to incubate over time for 3 days, I took a look at it by NMR, and I could identify peaks that corresponded to both ribose and arabinose. Now, there are hypothetically more than these possible peaks that could form in terms of all the possible different configurations of ribose and arabinose, including the furanose, pyranose forms, the alpha-beta anomers. So some of those peaks are present, some of those are not present in high enough concentrations because there is a dynamic equilibrium between those that prefers a certain configuration for those sugars. So the result of this is that Ribulos does readily isomerize in solution into ribose and arabinose. The next experiment that I performed was one where I dried down Ribulos with triaminopirimidine, and I wanted to see if I could form different nucleosides, both arabinose and ribonucleosides. And so I performed this reaction. These are the possible linkages that could form with these nucleosides, and I've omitted the pyranose forms, but those could possibly exist as well. And after I performed this reaction, I looked at it via NMR, and I was able to identify peaks that not only corresponded to the isomerized sugars, ribose and arabinose here, another arabinose peak here, but I was also able to identify peaks that corresponded to nucleosides of TAP and these sugars. So the result here is that it's pretty clear that Ribulos does form these aldonucleosides when it's reacted with triaminopirimidine. So another experiment that I performed then was basically I wanted to take each of these sugars and incubate them over time to see if I could get sort of a long-term isomerization rate. Once I incubated them, I would take time points every day, and then I would react those time points with barbituric acid at room temperature. Now, barbituric acid was useful in this experiment for 2 reasons. One, it is quite visible via UV, so it allowed us to monitor the progress of these reactions or the amount of nucleoside formed when we analyzed it by LCMS. And the other benefit of using barbituric acid is highly reactive. So it's one of the most reactive nucleobases that we work with. And so I could perform this reaction at room temperature, which means once it's heating up and isomerizing at a higher temperature, if I bring it down to room temperature, presumably it sort of freezes the isomerization state of those sugars, locks in the different concentrations of ribose or arabinose or ribulose, and then it will react with the barbituric acid, form those nucleosides, and then I can track them. So the results of this experiment show that this green trace is ribose, this blue trace is arabinose, just reacted with barbituric acid after being allowed to incubate for just a day. And then the 2 red traces are Ribulos. The first one is Ribulos incubated for 1 day, and then the darker red trace is Ribulos incubated for 408 hours, and these are all at 65 degrees. And once I react each of those time points with barbituric acid, you can see them here. Now, I have scaled these up. The yield of the nucleoside formation is small, so I've scaled up so that you can see the formation of these peaks. And what you can see is that the formation of the riboside peaks are increasing as you allow Ribulos to isomerize over time, And you can kind of also see the formation of this small arabinoside peak as well. So the result of this is that once you allow Ribulos to isomerize in solution, it can go to either ribose or arabinose, which then, once I add barbituric acid, will react to form these nucleosides. I did the same experiment with hexose sugars just to see if these trends followed based on the fact that they're ketoses and didn't have any effect based on the carbon number. And you can see the same thing here. So the green is glucose, not incubated at all, reacted with barbituric acid. The blue trace is mannose, again, not incubated for any time, reacted with barbituric acid. So you can see that there's a large peak here that represents a glucoside or a glucose nucleoside, and there's a large peak here that represents a mannoside or a mannose nucleoside. And when I incubate fructose for 408 hours, you can see it go from having no nucleoside peaks to having large mannose and glucoside peaks. So this, again, to me indicates that when you have these ketose sugars and you let them incubate over time, they can interconvert into the different aldose isomers, and those can then react with nucleosides or with nucleobases to form nucleosides. So the scheme that I'd like to leave you with is one where you have something like Ribulos, for example, which not only can interconvert between sugars, but these sugars go between their linear and cyclic forms. So you could have Ribulos that can interconvert to ribose and arabinose as well, and these can again also interconvert with their cyclic forms. But not only do you have this system, which I've kind of demonstrated, you could hypothetically have a larger system where you could have Ribulos interconverting into xylulose by the carbonyl or the formation of an enol in between the 2 and 3 carbons rather than the 1 and 2 carbons. And then that could isomerize into the sugars lixose and xylose. So in terms of the prebiotic context and implications of this, I think that once you form these sugars, they can isomerize readily, and they can basically interconvert into any possible sugar of that structural arrangement and length. So once you have an unbranched 5-carbon sugar in solution, hypothetically it could isomerize into any other unbranched 5-carbon sugar. So in terms of what this means for prebiotic chemistry, I think that these sugar-forming reactions don't necessarily need to be selective for something like ribose. If you can form Ribulos or any other 5-carbon sugar, then you're pretty much there. You're able to get any other 5-carbon sugar in solution. So in conclusion, I think the prebiotic formation of sugars likely allowed for some interconversion that gets you all these different isomers. You can have these nucleobases in the solution that would be able to sort of trap these sugars in their aldose forms, allowing for the formation of nucleosides, and that life or chemical evolution could have selected from this large pool of nucleosides on the early Earth. So I'd like to thank my advisor, Nick Hudd, and my group members, especially David Fialo, for helping me out with this. Several other collaborators, including Dr. Krishnamurthy, Dr. Minor Savan, Dr. Schuster, and the rest of my graduate committee have been a great help with this, and I'd like to thank the NSF and NASA for funding. Thanks for listening.
Very nice. Do we have one question? One quick question? Yes, right here.
So what actually drives that nucleosidation reaction that also probably has like a pretty strong kinetic barrier?
So it's not irreversible. You could have nucleosidation and then hydrolysis. You can imagine, this is all done in solution phase, but you can imagine some sort of system where there's also a drying cycle, which it would allow for that nucleoside formation to happen. And then the other component that could drive that nucleosidation in sort of a directed fashion is, I mentioned that these nucleobases assemble in a supramolecular fashion. So you could imagine that perhaps when they react with a sugar, they could then be drawn into the supramolecular assembly where the sugar is hanging off the end, and then perhaps you could have some oligomerization followed, once you get a critical mass of sugars or something like that.
Very nice. Thank you much, Tyler.
Thanks.